Hello. So this week I will be doing my mini lesson, and this is my boyfriend Matt. He's going to be the student, and we're going to be learning about mitosis. I just wanted to quickly describe the aspects from the rubric that I needed to include in this. So my vocabulary piece will be mostly learning the vocabulary during the modeling activity. And then I will show a short video at the beginning that is how the vocabulary is introduced. And then in addition to that, there's several vocabulary words that we don't cover because I would have taught those in the lesson before, because otherwise the topic has too many new words to teach in one lesson. So I wanted to stick to the 5 to 7 range, and to do that, it has to be kind of assumed that the students have learned some of the words earlier. So that's the vocabulary, and then my language function is, there's two actually. It's describe and model. So the students will be describing mitosis orally and also written, and then they will be modeling mitosis using these little cooking string twine or whatever. So that will be the language function. And then my syntax is a mnemonic device that I'll be teaching to Matt to help him remember the order of the steps. And then the discourse will just be discussion between the teacher and the student. And for the modeling activity, I'm going to sort of pretend to be his partner because there's only one of him, and in my lesson the two partners are like talking for the whole part, so I'm going to be taking on that role. And the one last thing to add is I'm not going to play the video because that would be strange for you guys to watch me watch a video, so I'm just going to link that in my Canvas post. So I already showed Matt the video, and when I get to that piece I'll just mention it and then we'll move forward. Okay, ready?
Ready.
Okay, so we're going to be learning about mitosis. And first I want to start off with the learning targets. So I want you to know what we're going to be doing. So I can describe the steps of mitosis, written and orally, and I can model the steps of mitosis. So I'd like you to write down the learning targets in your own words, or you can copy them if you need to see them. They're on this piece of paper.
So I can describe, written and orally, the steps of mitosis, as well as model them.
Yes. And do you have any questions about the learning targets, or do you feel pretty good?
Let's jump into it.
Okay. So this is when I would play the YouTube video. And after we would just watch the video. Okay, so we will be modeling the stages of mitosis, and you just learned about those briefly from the video. And so they're interphase, prophase, metaphase, anaphase, and telophase. Can be a little bit confusing. There's a lot of them. So I have a helpful way of remembering the order in which they occur, and that is using a mnemonic device. So the one that I like to use is I picked my apples today. So each letter corresponds. And if you, I don't know, is there one that you can think of?
So the order is IPMAT. I
Yes.
picked my apples today. I think I can remember that. I like it.
Cool. Okay, so when you're good there.
Yep.
Now we're going to work on our KWL chart. So we've done these before in class. So what about, just blurt things out to me, I can write them down. What do you want to, or what do you already know about mitosis?
It's the splitting of cells.
Okay.
The process is made up of 5 steps, which I want to know those 5 steps a little better.
So you want to know like what happens at each step?
Yeah.
Okay. what about, do you know about why it happens, or would you like to know about something like along those lines?
What triggers it, right?
What triggers it?
I think that'd be a good thing to know.
Okay. Okay, cool. So we're gonna revisit this after we've completed the activity. And now we are going to start the activity where we're modeling and describing the steps of mitosis. And I want to be really clear about what exactly that means when we're modeling and describing. So we're going to have this handout somewhere. We're going to have the handout, and the handout here tells you what happens at each stage. So we're going to identify the stages. Then we're going to record each step onto our worksheet. And then we're also going to talk about it. We're going to communicate verbally. So that's how we're going to describe it. And then to model, we're going to be simultaneously modeling and describing. And so, again, we identify the stages of mitosis using the handout. And then we're going to manipulate the string that symbolize the chromosomes. And that's going to mimic each step. So we're going to be modeling it in front of us, working together. And then as we do that, we're going to sketch each step. And that will be how we model and sketch. So we can start, and then I have this paper for you. We're going to be this is his worksheet, so we're just going to be drawing them on here. And then here's our handout that has the descriptions of each step. So, and I will be his partner for this part. So first we have interphase. Do you want to take turns like reading out loud maybe?
Yeah. So the first step of mitosis is interphase, where chromosomes duplicate and the copies remain attached to each other.
So with these model chromosomes, it's already duplicated. So these are exactly identical. There's two, they're called chromatid, sister chromatid.
These being each length of string in this case?
Yep.
There's a chromatid?
Yeah, and so together they make the chromosome, so it's duplicated.
So these are post-interphase?
Yeah, so this basically is the product of interphase, is the duplicated. And it looks like if we look at the picture, they're all just floating around in the center of the nucleus.
Okay.
So if you want to sketch those, and you can just draw them as an X.
Okay.
That's easy to do that.
So draw them after they're duplicated.
Yeah, so or we want to draw what we see in front of us. So just kind of, they're all just floating around in there. There's not really rhyme or reason to it yet.
Okay.
Perfect, that looks great. And I'll read this one. So now we're moving to prophase. In the nucleus, chromosomes condense and become visible in the cytoplasm and the spindle forms. So in interphase, they're a little bit looser, and then when you move to prophase, they condense. They're more defined. You can see the shape of them more.
Shape of the chromosomes?
Yeah. Before it was kind of just long coils of DNA, and now they're tightly stacked. Yeah. And they're still floating. So this one looks pretty much the same.
But just condensed a bit more.
Yeah.
A bit more defined.
I would maybe make
Yeah.
these a little bit like longer. Yeah. Perfect.
Works.
And then we have metaphase. So the nuclear membrane breaks apart and the spindle starts to interact with the chromosomes. The copied chromosomes align in the middle of the spindle.
So you have prometaphase and you've got metaphase. What's the difference there?
It's basically like the start of metaphase. Okay. So we're just going to draw them as one step.
Okay. Right, because it's IPMAT, not IPPMAT.
Yeah. So one way I like to think of metaphase too is M for metaphase, M for middle. So this is when the chromosomes are lined up in the equator of the cell.
Okay.
And the spindle fibers are what pulls them apart, and that forms at each pole. So if we want to model it, it looks like this with all of them in the middle.
Okay, so they're all lining up down the center because they're going to be pulled from either end,
Yeah,
like we
and
saw
they're
in the
like-
video.
And they're going to be pulled
This way.
so you want them to be lined up in the sense that they're going to be pulled that way, not vertically but horizontally. Does that make sense?
They're going to be pulled perpendicularly to the direction that they're aligned.
Yeah.
Okay.
Yeah.
Yes, that makes sense.
So if we want to draw that, perfect. And then the spindle is basically these little fibers that come down from the poles of the cell. And that's
Okay.
what pulls the chromosomes apart.
Got it.
And now we have anaphase. So do you want to read anaphase?
Yeah. Chromosomes separate into two genetically identical groups and move to opposite ends of the spindle.
Yep.
Okay. So now we
So now
start tugging.
this is when they separate, because currently there's too much genetic information in this cell. It's
Yep.
twice the amount. So we have to pull them apart, and then they'll be the two pieces in each cell.
So this is where the chromosomes will actually split.
So, yeah, this is when they actually get pulled
Okay.
apart, and you can see in the picture the spindle fibers are pulling them.
Okay.
Good.
Got it. So it looks something like this then, where I'm drawing the pole on either end
Mm-hmm.
and each chromatid being pulled away from the center, completely separated.
Yep.
Okay.
And then you would draw the little they're kind of bent because they're being
okay.
tugged. So those are the actual fibers that are tugging at the.
And then so our string is what's represented by this V at the end of the.
Yep, exactly.
Okay.
Okay, and the last phase, telophase. Oops, typo. I can read that one too. So nuclear membranes form around each of the two sets of chromosomes. The chromosomes begin to spread out and the spindle begins to break down. So currently, or throughout this whole process up until now, this has been inside the nuclear envelope, or the nuclear envelope that contains the genetic information has dissolved, and now it's forming again, but it needs to form two envelopes around each of the sets. So you can see it pinching in here, forming the new
Okay.
right around the chromosomes. And then this is the actual cell that's pinching in.
So then at this point it looks like the inside of the membrane is similar to that of interphase in that they're still a bit chaotic within each.
It's
membrane.
returning back to normal, basically.
To its original state.
Exactly, yeah. But obviously with two daughter cells. Perfect.
Okay.
And the last stage, which is technically not mitosis, because mitosis is the division of the nucleus,
Mm-hmm.
and cytokinesis is the division of the cytoplasm.
Mm-hmm.
And that's our last stage, which is just the final pinching in around each of the nuclei.
Cool.
So the cell splits into two daughter cells, each with the same number of chromosomes as the parent. In humans, such cells have two copies of 23 chromosomes and are called diploid.
Got it.
Yeah. And
so cytokinesis is not actually part of mitosis.
it
always
it's just
preceded.
it's what's after mitosis.
Yeah.
So first the nucleus divides, and then the cytoplasm divides. So it always does happen when you have mitosis. So I would say it is a part of it, but technically it is like
It's
a
not
different.
one of the 4 steps.
Yeah.
Okay.
Or it's not the division of the nucleus. It's its own thing.
Got it.
When the
Got
cytoplasm divides.
Okay.
Okay, so we got that. We modeled, and I maybe really quickly just say like one main point about each stage.
Sure. So interphase, still a bit chaotic within the nucleus until prophase, where we'll say the chromosomes start to tighten up. Some order is seen within the nucleus. At the metaphase is when they will begin to align or prepare for the actual separation of the chromosomes, at which point anaphase is where the chromosomes will split into the individual chromatids, until finally we reach telophase, where the nuclear membranes, the new nuclear membranes, begin to form around each daughter cell as the nuclear envelope begins to split of the original cell, and then cytokinesis is where they actually
Perfect.
snip.
Yeah, I feel like you got it.
Got it.
So, how do you feel about the learning targets after doing the activity?
Yes. Now I understand it quite a bit more. I can describe it written, orally, and via the model.
Cool. Okay, well, I think we are going over our limit. So that is all I'm going to show right now. And to do the full lesson, I would return back to the KWL chart. I would also have some sharing out, hearing some student voice, potentially on a whiteboard, having the groups each draw a different stage, and then show the class and describe. So that's what I would want to do for that. And then go back to the KWL chart and talk about what everyone has learned, and then also revisit the learning targets again. So yeah, that is my mini lesson, and thank you, Matt, for being my guinea pig, and I will see you guys later.